everyone. Thank you for joining the first ABI seminar of the year. We have this great pleasure to have Dr. Naz Ebrahimi giving us the first PhD completion seminar of the year. Many of us know Naz. She joined the ABI in 2013 after completing bachelor's degree in biology and a master's in genetics from Iran, and she completed her master's at the University of Oxford, and then he joined the ABI and did PhD with the Peter, Chris, and others. So she built, designed and built a comprehensive experimental and computational method to investigate the early heart development, and she will be discussing that with us today. Her seminar is entitled How the Heart Grows, from multiscale data to a multiscale model. Over to you, Naz.
Thank you. Thanks, Vicky. So thanks all for coming to my talk. So as Vicky mentioned, I'm going to talk about my work, which I did for my PhD, how the heart grows from multiscale data to a multiscale model. I want to start with acknowledgements. Many thanks to my supervisors, Peter Hunter, my main supervisor, and my co-supervisors, Chris Bradley, Fabiana Kuke, and also my colleague who supervised me for the first 3 years, and I never forget his help. My work was funded by Marsden, and I have a special thanks to Dane at the ABI's Microcity Imaging Center, also Biru at the medical faculty. Actually, I've got a very long list of amazing people, colleagues and friends and staff at the ABI and the University of Auckland, which I can't name everyone here, but thanks to all. So let's start. I'm going to go through the development of heart, which is the subject of my work. For those which is not familiar with the field, just to get a brief overview of that. So this diagram shows a very simplified cartoon of the development of the heart in rat, but it is a very conserved process in vertebrates. So if I say just simply, we have precardiac cells. Through the gastrulation, they move anterolaterally to the cranial or anterior side of the embryo and make 2 heart fields up there. And then the reason that we divide them into 2 heart fields because they have distinction in the time of formation, and also they give rise to different structures of the heart, which is color-coded here. So the first heart field, which is colored in purple, formed a horseshoe-shaped structure called cardiac crescent. You can see in figure B. And then the second heart field positions medially to the cardiac crescent. And then later cardiac crescent fused to make a straight structure, straight tube. We call it a straight heart tube. But it's not actually a closed tube. The cross-section in the zoomed area, which shows the in figure C, shows that the tube is open at the dorsal wall of the embryo, and it is connected as a kind of open tube all along the heart tube, connected to the embryo through a structure called dorsal mesocardium. I will use this dorsal mesocardium later, that's why I named it. But in terms of the heart, at this stage, we have the myocardial layer, we have forming endocardium, and between myocardial layer and endocardial layer, we have a thick extracellular matrix, which is acellular called cardiac jelly. And then in figure D, you see that tube bends ventrally and shows a rightward rotation. which to form a structure called C-looped heart. When you look at it, you can see it resembled a letter C. That's why it is called C-looping phase or C-looped heart at this stage. Then heart shows more rotational movements called and form a S-looped heart, and then through more morphological changes, which is a very complex process, goes through the septation and valve formation and etc. to form a 4-chambered heart. In this slide, it shows a very nice electron microscopy image of the heart during the development in Czech embryo. And as you see, we have the same process starting from heart tube formation, going through C-looping phase, S-looping phase, and to form the 4-chambered heart. My work is focused on the C-looping only. And when we talk about C-looping, there are 2 aspects. A, the rotation of the heart through the right side of the embryo, and B, the directionality of the looping toward the right or left. I have to note that my work is focused only on the rotation, how the heart shows that ventral bending and rightward rotation. And before going further into C-looping, maybe I should answer this question: why studying C-looping or looping? Looping is a very crucial stage in the development of heart because it is essential to bring the tracts and chambers with respect to each other and form the basic geometry which is needed for the 4-chambered heart. And abnormal looping could result in various congenital heart diseases, like these 2 examples here: double outlet right ventricle, double inlet left ventricles, and other alignment defects. That's why many studies have been done on the investigating how the looping happens. Okay, when it comes to C-looping, there are different configurational changes. As I said, it's not only rightward rotation, it has ventral bending. Although through the time, investigators found it, for them, the looping was, at first, the looping was just simple winding to the right, and then they realized that, no, it's actually C-looping and S-looping, so it has 2 components. And further morphological investigation showed that C-looping itself consists of 2 phases: ventral bending and rightward rotation. And even after knowing about this ventral bending, at first we had a 2D concept about the C-looping. In 2D concept, this bending happens in the 2D plane. And as a result, the right and left side of the heart before looping is corresponding to the left and right side of the heart after looping. But later, they found that this rotation and bending happens in a 3D space, and we have to look at it three-dimensionally. In 3D concept, as you can see in the figure bottom panel, the dotted line shows the ventral line, the middle of the heart, and we can see as the heart grows and loops, the middle part of the heart, which used to be, the right and left side of the heart in the straight tube, after looping, located in the ventral and dorsal part of the looped heart. So it is very important to study C-looping 3-dimensionally. In terms of mechanisms control looping or C-looping, I made this diagram just to make it simple. There are different hypotheses. If we imagine a straight heart tube as a just simple cylinder, we have the midline again, right and left side of the tube, and the cells are shown by the hexagons. We have same number of cells with the same size at two sides. And one of the first hypotheses in the field was buckling. So if we imagine this tube is fixed on the cranial and caudal part, and if the heart grows, in length, grows in length, but it is fixed at top and bottom, and then the heart bends. So some studies contradicted this buckling theory because they showed that they could grow hearts in vitro in the lab, but actually that looping is simpler than the actual looping in vivo. And again, in 2014, another study bring this one up again, and they use some modeling to show that the growth-induced buckling could be a driver of the C-looping and S-looping, but still they couldn't answer how the ventral bending happens. They couldn't simulate that. Another hypothesis in the field is differential growth. So if we imagine the cells grow more in number in one side than the other side, it will result in heart to bend. And the growth could be in terms of the increasing in the number of cells or in the size of the cells. This growth this differential growth could be driver of the bending. This is actually the most accepted hypothesis in the field at the moment, but there are many studies that could provide evidence, but there was a very recent study published this year. They argued that differential growth is not the main driver, and they introduced cell rearrangements, but still differential growth is considered as a main driver. So I can discuss about that paper later through my talk as well. They took completely different approach from these works. Another hypothesis is cell shape change. Well, again, if we imagine the cells align longitudinally in one side and circumferentially in another side, it could help the heart to bend. Cell shape change is not considered as a main driver in the field, but it hasn't been contradicted. So they consider this mechanism as a minor player in C-looping. And another example is cardiac jelly swelling and dorsal mesocardium tension. As I said, the heart was attached to the dorsal mesocardium, and it makes that side of the heart stiffer than the other side. And if the cardiac jelly swelling happens during looping, which happens, it causes the heart bend in the ventral side. These are the kind of morphomechanical forces that could drive the looping, but we also have morphogenetic factors as well. We know that looping is controlled by a very complex regulatory networks of the transcription factors and then the signaling pathways and the genes. All these complex network control the cellular responses, and in turn they could control the deformation during C-looping. But it is still unclear that how these known factors, subcellular factors, could control the cell responses in order to drive the C-looping. We're missing that. Based on this information, the literature that I did for my PhD, then I decided to look at the early development as a developmental system, as a multi-scale, multi-level system. We have different scales in developmental system. There are interactions between scales and within scales between the heterogeneous components and each scales. And these interactions are not linear. And most importantly, we're dealing with emergent behavior of these components. By emergent behavior, I mean the result of one level doesn't come from the individual parts of that network, the collective effect of the components could produce the outcome of that level. So therefore, I decided to take a multi-scale approach to look at the sea looping. And because we were missing data, and it is very important to have a very synergistic collaboration between experimental and computational works, I decided to do both experimentation and computation for my study. I start with experimentation. I have to be a bit quick here. So the aim of this part was to develop a pipeline to obtain a three-dimensional multi-scale data from a single embryo at different biological scales, from cell to organism. And because we wanted to look at the dynamic of sea looping, we wanted to have data from different time points during sea looping. So I use both Czech embryo and rat embryo as model organisms. This slide shows a workflow for the experimentation. Basically, I combine two imaging modalities of confocal and micro-CT imaging to be able to capture different data sets at different biological scales. Briefly, if I can go through it, I took the embryo, stained it for the whole amount confocal imaging, and then because we wanted to keep the heart intact, and I had to avoid physical sectioning for image. And did micro-CT imaging on the whole embryo. So as a result, I had two data sets from each embryo. One confocal, which could cover cell to tissue to organ level, and the other one from the micro-CT, which was covering from tissue to the entire organisms level. Going through the results, so the aim was to start from cell level. So my plan was to stain myocardial cells and stain the cell boundaries to be able to segment individual myocardial cells for further analysis. I used triple staining. If you look at so it shows a part of the heart embryo from heart from the Czech embryo. And in the zoom area, top left shows the multi-channel. So I have the red signal, which shows the myocardial specific signal. So I can say it's myocardial. I have nuclei, which is stain. I have DAPI, which is stain nuclei to be able to distinguish between cells with intercellular space cells if needed. But the problem was in top right, you see that that myocardial specific cell membrane stain was failed to stain the cell membrane when the cells were exposing the cavity, either the cardiac cavity or the cardiac jelly. So I got signal when the cells are attached together. Therefore, I had to use WGA, which is a global cell membrane stain, to produce a complementary signal with the myocardial stain. So in bottom right, I can see that I got a very nice signals around the myocardial layers from WGA and the red signal. And that red signal helps me to distinguish between these two layers, which one is myocardial layer, which one is not, which is really hard if you work with confocal image. It's not possible if you don't have any clue. So for the Czech embryo, I couldn't fit the entire heart in one tile. So I had to use either 2 tile or 4 tiles. I used tiling, I imaged the whole heart with overlapping tiles, and then using stitching, I was able to stitch this image and get the image of the whole heart. And then 3D rendering in figure C, you can see the whole heart from different channels. I got the same result for rat embryo. For rat embryo, I didn't have to do a stitching because I could fit the whole heart within one type. And then exact same embryo, I took them for the micro-CT imaging. So these are the results for the Czech and rat embryo. It involved lots of preliminary work. I don't have time to go through that. I can show the ultimate results. So it was really hard working with these delicate samples, and we wanted to manage to go down to the sub-micron resolution. So I've got a video. I played the video. This is the 3D micro-CT of Czech embryo. The whole embryo is imaged at 1.5, but I had another set of data set from heart, which is down to 0.5 micrometer resolution. And even with this 1.5 resolution, you can see that we are completely able to resolve single cell layers like forming endocardium, as I label here, and myocardium. The heart showed collapse, but it's because of the contrast agent solution, and it could be fixed later, but it was out of the scope of my study to work on that. So from the experimentation, I got two data sets from confocal and micro-CT, both 3D coming from the same embryo. So I have cell to whole organism level data set for each embryo. Then I moved to the computation part. So I had basically three aims: to generate shape models of the C-looping hearts, which was anatomically accurate. And then the second was to segment individual myocardial cells within the entire heart, measure the cellular features, and investigate the spatio-temporal of those features within the heart. And third one, to quantify growth at both cell level and tissue level, and address how these two data sets, how these two levels are correlated. I mean, how tissue level deformation is associated or correlated with the changes that we see in cellular features. First part of the computation workflow, so I got those two data sets. What I did, I used a 3D confocal data set to segment myocardial cells. And then I used this data set for anatomical landmarking, temporal ordering of my samples, and spatial registration and alignment of samples. We should remember that I'm going to get samples from different time points. So there are different samples. I have to register them and I have to order them temporally, because in Czech embryo, it's kind of impossible to define the exact developmental stage. And the difference between the time points, it's about an hour, so maybe less than hours. So I use this data set to comprehensively look at these kind of pre-processing. And then I use the segmented myocardial layer to generate the geometrical model of each heart. So at the end, I expected to have an anatomically based geometric model of heart capturing both spatial and temporal dynamic of C-looping. How I did it, I selected 4 chicken embryo at different stage. By just examination under the light microscopy, I could kind of guess that they are within the C-looping, but I couldn't order them temporally because they are very close. And then I developed a semi-automatic workflow in a software called Amira to segment myocardial layer. And it's nice to show it here, like without, as I said, having the myocardial specific stain was very important. Like these figures shows the image from the top part of the heart, and in the left one, WGA, where all the cells are stained, it's really hard to say which cells are part of the myocardial cells. Even the combination of WGA and NCAM1, still hard. But when I have NCAM1, I can be sure what part is myocardium, and then I use this one as the mask to segment the entire myocardial layer, which is of our interest. And then with this method, I successfully segmented the heart, which is, as you can see, it is kind of sandwiched between different layers. And it's really hard to segment that. But using that stain, it was fairly easy to segment heart. It's a very precise segmentation. And then the next step was anatomical landmarking. Again, the hearts in the right-hand side, there are 4 samples, the confocal images of my 4 samples. We have the time point 1 heart tube to the time point 4. And it is really hard to find the corresponding region. They are coming from different samples and we don't have any cell tracking or anything. So that's why I use literature. I use different landmarks to find the corresponding regions. Also, I segmented endocardium in the left panel, you see, because using endocardium, I can find the ventral point and dorsal point of the heart throughout the heart tube, which was very helpful. And then I did a spatial registration because, again, the heart's image at different coordinate systems, so bringing them. So I use rigid registration in EMIRA to align all the hearts. And then for temporal staging, I use different approaches. For instance, the level of the rotation of the heart would help to see, because we know that the heart is going to rotate. So even after spatial registration, I could say qualitatively which one is first and then next. But I did I took more quantitative approach to confirm that. I used matricity data set and did different measurements in terms of rotation or the level of detachment of the heart from the dorsal wall to order the samples. So I was confident that I got the right order for my samples. And then I used the generated point cloud from the segmented myocardium, which basically represent the heart geometry, to fit it onto the template mesh in OpenCMS, a finite element mesh. And then I use, for instance, I use those anatomical landmarking to fix the points because I want to find the corresponding region between 4 time points. And also I use endocardium to align the heart properly. And then through various iteration during fitting, at the end, I could generate a very nice and smooth mesh of and model of the heart. And here's the result for the 4 hearts, time point 1 to 4, from a straight heart tube to C-looped heart, and also the 4 overlaid meshes that shows the dynamics of the C-looping. I think I have another video to show just as a break. So it's just an overview. Again, I have 3D confocal images of the heart. This one is time point 2, and then the 3D rendering of that confocal image. Then I segmented my myocardial layer. Generated a 3D geometry of the heart, and then got the point cloud out of it. And then fitting to a finite element mesh. Point cloud for the myocardium, and then the blue one shows the point cloud for the endocardium. And finally getting into a nice smooth geometric model of these time points. And these are 4 time points. Okay, moving to the next part of the workflow. So again, 3D confocal. So the next, so I met the first goal. The second one was to segment individual myocardial cells and then extract the cellular features and then map those cellular features onto the geometric model we just generated in order to produce a data set which has the heart with embedded cells and cell features and capturing the spatial and temporal aspects of these features. So for cell segmentation, I think it was one of the hardest part of the study. I tried all the algorithms and I found it impossible. So at the end, I had to try machine learning techniques. So from A, I start from the beginning. So I had the heart, I have 3 channels in B. The images are big, so you can't fit these big images, 2000 by 2000 pixel into the computer, fit into the computer because of the memory problem. So I had to develop a kind of pipeline where I get the big images. I use a striding and slicing technique where you develop the the pipeline. You take your images and then you divide it into very small pieces of images with overlapping, and then you feed those small images into your convolutional neural network. And then once you have the output from the segmentation, you kind of merge all those small images back again together to make the original image of the heart, which shows in G. I have the heart with all the cells segmented. So for this part of work, I had to do a bit of manual segmentation to provide training set for the unit, the convolutional neural network, which I used, but it wasn't that much. So the result was amazing. It was efficient. With manual segmentation, it was impossible. I could manage to segment 100 cells per day, but with this technique, 100 cells took 8 seconds or less. So it was impossible to segment all the cells within the heart with more than, I don't know, 5,000 up to 18,000 cells within the heart. The machine learning was more accurate and robust than the manual segmentation. The image shows the results from the, like if you look at the first 2 columns in the right, the prediction result is better, smoother, less noisy, and more consistent. And this one shows a 3D image of the manual segmentation and the model prediction on the same image stack, and you can see it is much smoother, less noise. And most importantly, with this method, I was able to get the individual cells segmented within the full heart. So again, there's a very short video that shows the heart with the individual cells segmented. I have overlaid the segmented cells over the confocal image. Okay, I have to skip. I have to be quick. So basically what I had to do now, I had the segmented cells, the blue one in the right-hand side. You can see here, I used Amira. In Amira, we treated individual cells as one entity, and then I ran many analyses to measure different cellular features. And of course, for the cell level, I made sure that not only at tissue level, but also at the cell level, the cells are aligned with respect to both global and local coordinates, because we were looking at the orientation and it was very important. Then I had to map the cells with their cellular feature values onto the surface of my mesh. And I took two different approaches for mapping. The first one, projected cell data, shows the cells mapped onto the surface. Each glyph shows actually individual cells, and white to red shows the value for that specific cell feature. For instance, here is cell volume. Field fitting approach was to, as you see for the raw data, it's hard to see the pattern. So with field fitting, we kind of treat the cell feature as a field, and we get a nice mean value overlaid on the surface. So it's easier to capture any existing pattern. So I explain more in this video because in this figure, it's really hard to see, but in video, it's very nice. So this example is the cellular feature, right? The entire cells are mapped onto the surface. And then in some parts, we see that the cells shows a very various value. They are heterogeneous. But in the other parts of the heart, we have some areas that shows a more homogeneous population of cells, like this area. And then same data set used to fit the volume data over the surface. And again, we kind of see those patterns in this one as well. But I wanted to have both data sets for my analysis. Like here for cell orientation, again, we have single cells. The glyph shows cells. The orientation shows cell orientation, and the color shows the magnitude of the stretch or elongation of the cells. And in this region, for instance, they more aligned horizontally, but there are some other parts of the heart that the cells are completely random in terms of the alignment. Again, field fitting helps to capture all those patterns easier, but here glyphs doesn't correspond to single cells. It's just to show the orientation. Okay, so now I have a data set with all the cells and cellular features embedded within the heart with that spatiotemporal information. Then I could do analysis. I did lots of analysis. I don't have time to go through it. So I started with entire heart analysis. And even with this simple analysis, I found very interesting things. For instance, from the first step, I found that when we talk about the growth, it's very important not to look at only the growth of cells and the volume of cells, but the changes in the intercellular space volume is very important as well. And we captured a very nice information regarding that, which was quite new in the field. I'll probably talk about it later. I did temporal pattern using fitted field, spatial pattern analysis using fitted field, but they are more qualitatively, and we could see the patterns. And then to be more precise, I wanted to do quantitative analysis to be able to investigate differential growth hypothesis. In differential growth hypothesis, is based on the fact that cells in the ventral sides grow more in the dorsal side. That's why I chose 3 regions: outflow tract, dorsal, and ventral. And I took all the cells with their features within these 3 areas, and I ran lots of analysis to look at different features. For instance, I did regional spatial pattern analysis from the cell data and the data file. And as I said, it's a long analysis, so we don't have time to go through that. Also, I did the regional temporal pattern, then I could look at the changes between different time points at these 3 different regions for different features. At the end, I was able to kind of provide a comprehensive table of these spatial and temporal changes. But apart from that, we found a few things which was kind of very interesting and also new in the field. Differential growth is based on the study that it was the first time in the field that we had individual cells segmented and their volume was measured. In previous study, they stained nuclei within the entire myocardial layer and then divided the number of the nuclei with the over the tissue volume and estimated the average cells. So they're missing information about intercellular space information or volume changes, which is very important because we found that it could be a very important factor. It shows very interesting information. It shows a spatial regional pattern, and these patterns change through different phases of C-looping. And we could provide, based on this information, most of our studies were in agreement with differential growth and cell shape analysis. At some point, some part, it was a little bit of not contradiction, but different. And the reason is because for cell shape analysis, for instance, all the study comes from either 2D imaging, or if they've done the 3D imaging, they sampled just a few cells and did 2D analysis on the cell shape, but we did 3D shape analysis and all the imaging and analysis carried out in a 3-dimensional manner. So we confirmed differential growth, but we could provide more information with higher resolution, and also we could introduce new features to be investigated more in the field. Another interesting things we found, it was variance analysis. So when we look at the absolute value of the features, we couldn't find any similarity in the patterns coming from different cell features, and it was strange. And then we decided to look at the variance. By variance, we're looking at the difference between the value for one cell with the surrounding cells. And it was interesting because for different features, variance analysis showed similar pattern. Like here for cell volume in time point 2, you see at time point 1, we don't have any significant pattern. At time point 2, we can see in the outer curvature within that blue and green circles, you can see an area with low variance, and at the outer curvature in the green one, there's a region with high variance. And then these ring of low variance grow over the time, and the high variance area gets smaller. For the principal eigenvalue, which is related to the cell orientation, we see the same pattern. And again, interestingly, for the cell anisotropy, we see the similar but reverse, like the ring shows high variance and the outer curvature shows low variance. So we could connect, we can explain this pattern with other studies in the field, which is more about the functional aspects of the heart development. For instance, from the ballooning model in the development of heart, they say that precardial cells comes to the heart tube and they're not differentiated. In the outer curvature, there are some regions that shows pattern that the cells start differentiation into the functional myocardial cells. And the pattern from our study shows similar pattern with those. And it's very interesting. It shows the relationship between structural changes, cellular features, and functional features of the myocardial cells. But unfortunately, I couldn't work more because it was out of scope of my PhD. So the last part of the workflow, I think I have only 5 minutes. It's to, now I have those meshes, I have the spatial temporal data set. Now I want to use them to quantify growth at cell and tissue level, and then look at the changes of these cellular features and tissue growth within each time period, and then look at the correlation between them. So what I did to quantify growth, I use kinematic analysis using the first time point and second time point, and using this deformation to quantify the growth. So again, there is a video quickly going through that and explaining what I did. So using kinematic analysis, we could quantify the magnitude of growth and also the orientation of that. Our results in general was in agreement with previous studies, but again, provided more localized and quantified information. For instance, here, when the heart bends, we see the outflow tract elongates, the ventral sides elongates, but not the dorsal side. In the next time period, this one, where the heart rotates toward the right, we see that the outflow tract doesn't grow, doesn't elongate. And interestingly, the ventral side doesn't elongate, it shows elongation. And we found it very interesting because the right side of the anterior side of the heart shows higher elongation than the other side in the ventral. And it could be related to the rotation part, because we know in the first time period, heart just bend ventrally. But in the second time period, instead of bending, it shows rightward rotation. So there are two different components. And then in the last time period, where the heart forms the C-looped heart, again, we have different profile for the growth at different regions. And it is very interesting because they believe that the different components of the heart controlled with different mechanism, and we could quantify that and especially show how the growth changes during the looping. So to quantify growth at cell level, it was easy because we had all the cells with embedded features. So we could measure the changes between time points. And then we use general linear model to look at the correlation between the tissue growth and changes in cellular features. And we found that the tissue growth is correlated to the changes in number of cells, volume of cells, cell anisotropy, and changes in the volume of the intercellular spaces. And adding the time and region to the model significantly improved the model. And it's very interesting because it shows that the spatial and temporal aspects of the growth is very important. In terms of the orientation, the result was interesting. Correlation between tissue
Sorry, I think there is a problem with the Naz's connection here. She was cut off. Let's give her a few minutes to come back here and then finish her talk. I think we're really close to the end. I'll just send an email.
Sorry, guys.
Good.
That's my connection. So.
Okay.
Okay, can you see my screen?
Yep.
Okay, so there are lots of spaces to improve this workflow. We can add more number of samples. We can improve imaging techniques. We can integrate this data into more mechanical modeling. And also look at the earlier stages. We can add subcellular level to this multi-scale approach. Actually, we introduce a very basic multi-scale model from subcellular to the tissue level, but it's just a preliminary work. So on the right, you can see the whole workflow, experimentation and computation. So I can say I could successfully develop the pipeline, both experimentally and computationally, to study early heart development at multi-scale levels. And we provide, for the first time, the 3D dataset of C-looping heart from cell to organism. And the dataset that have all the individual myocardial cells segmented, so researchers can do more research on that. It will be publicly available. And we could contribute to the differential growth hypothesis with providing more evidence, especially and temporally, while introducing new candidates for this field. Yep. So I really enjoyed my PhD, although it took longer than I expected, almost twice the time. And but I really, really enjoyed. I hope you enjoyed the talk. Thank you.
Thank you, Naz, for this great presentation. I have to say this is one of the most comprehensive PhD seminar that I've ever seen. It covered image processing, machine learning, finite elements, statistical analysis. So it covered pretty much all of the major areas in bioengineering. That's fantastic. And then, and look at this flow chart. Look at this. Wow.
That's why it took ages to finish.
Right. Great work. So this seminar is now open for question. Anybody who has a question to Naz, you can either put it on the chat box or just unmute yourself and ask. While people are thinking, one question that I had while looking at your seminar was because you had this very detailed data set from imaging, and then you were using field fitting to fit it to the finite element model. And in that way, you lose some of the data by doing field fitting because that involves some smoothing of the data set. So how much of the data was lost during that process?
Yeah, most of the analysis I did, I used raw data. So I was concerned about losing that data. All that diagrams that I showed, I had a file including all the cells with all the measured features. So I used field fitting just to kind of start with qualitative analysis. For instance, without that field fitting, maybe it wasn't easy to capture that variance. Pattern from variance analysis. Once I had that, and then I knew where I have to look at, and then I have a big dictionary made for my raw data, I could pull out that population of cells and do the statistical analysis on that.
Okay. Yeah, great work. Anybody else who has, you have a lot of people congratulating you in the chat box. Marty says,
Thank you all.
comprehensive and excellent tour de force. Great work, amazing work, yeah. So yeah, so this seminar will be uploaded to the Cassini site, where you will have chance to ask Nat some more questions for the next 48 hours. Sorry, Nat, to have to bother you for another 48 hours.
No, we'd be more than happy. Please ask questions.
Yeah. So so go back. If you click that link, that will lead to the this page, and then there is this video and Q&A section, which will start after the seminar. So you can continue to ask Nat some more questions if you have one. But if there's no other question, maybe the people are still thinking. I think it's such a huge topic that it takes some time to digest and think about it. But yeah, this will be made available.
I had to go over it very fast. Sorry, I couldn't.
Go right. Detail. Yeah. Okay. That's okay. That's okay. We understand that this is such a lot of material. So yep. Cool. So thank you, Nat, and thank you everyone for coming to the seminar today. And great work. Thank you again.
Thank you, Vicky. Thank you all.